everybody. My name is Brent Peltola. I am the Executive Director of Partners in Research Canada. Welcome to this PIR live event on synthetic biology with our guests Patrick Diep and Scott Proulx from BioZone at the University of Toronto. I'm going to give Patrick and Scott a minute to introduce themselves, and then we're going to go through their presentation, and we'll get to questions at the end. Please remember to open your Q&A window and submit questions in that way, and then I will relay those questions to Scott and Patrick as appropriate. So with no further ado, thank you, Scott and Patrick, for joining us today.
So my name is Patrick. So I'm a biochemist. I'm currently doing my PhD right now in chemical engineering. And I work with bacteria and trying to get the bacteria to soak up metals from wastewater in mines.
I'm Scott. I just recently finished my master's in metabolic engineering. So currently I'm working as a research associate, and my work involves mostly metabolic engineering of bacteria like E. coli to make non-natural chemicals.
Fabulous. So if you guys want to open your presentation, and we'll get through that.
There we go. Okay. Can you see?
There we go.
Okay. So we wanted to talk about synthetic biology and the future. And to kind of start this conversation, we want to introduce a concept that you might have heard before in your biology classes or some biology part of your science courses. And that is the idea of central dogma. Central dogma is an abstract concept, but very simply put, it is a flow of information that happens in all living things. And this flow of information typically goes from DNA up here down to RNA, which is this red strand here, which can then be converted into your proteins, which is these green little globules. This flow of information is important because it is what allows cells to figure out what they need to do. And what scientists have been doing, biologists have been doing, is kind of studying these different parts of biology individually. And by studying these different parts individually, for example, studying proteins individually, it's allowed scientists to learn a lot more about big things and big pictures. For example, scientists have been able to learn more about how disease is caused and therefore produce medicines for these diseases very specifically. Understanding how biology works at this scale also allows us to develop new crops and understand how these crops and livestock can be produced for specific effects, food and fermentation, as well as understanding ecosystems from the micro scale and all the way up to the macro scale, just by understanding central dogma and these different parts of biology. Now, one of the things I want to point out is that studying central dogma, studying biology at the molecular level, for the most part, this has been studied by molecular biologists in the field of science. Something that Scott and I felt that we didn't really learn in high school was the difference between science and applied science or engineering. So science is pretty much trying to understand natural phenomena, things we see in nature, in life. And sometimes these natural phenomena pose research problems that kind of pique our curiosity. But these problems aren't necessarily always affecting society at a whole. And so the difference between science and engineering that we see is that in engineering, you're trying to find solutions for these problems that do affect society. And so science and engineering are basically two sides of the same coin. The coin itself is our understanding of the world. And science is trying to find more of this knowledge, trying to understand this world better. Engineering is trying to take this knowledge and apply it so that we can kind of address the problems in society with solutions.
Now, if we think about applying sciences and we want to apply it to one specific science, let's focus on biology for a second. So thinking back to what Patrick was talking about the central dogma, recall he said we start at DNA, that codes for RNA, and the RNA is translated into proteins. Proteins do the stuff that cells do. So anything that you have expressed, for example, the color of your hair, that has a pigment that is made by a protein. So we have tools actually that we can edit DNA. And this is our toolbox basically so that we can change the way that proteins work. Let's say we have a chemical reaction, and some of you have taken some chemistry courses and you know that we could maybe react A and B together to make a product C. So enzymes are special proteins that can make these reactions happen at very low temperatures and very fast inside of a cell or inside of the human body. And so what we can do is we can edit DNA in a certain way, or maybe give the cell some DNA that it doesn't normally have to express this protein that will convert A and B to C. Alternatively, maybe we're interested in just producing the protein itself. An example of this would be, for example, insulin. Insulin is used to treat diabetes, and insulin is actually a protein. So what people have done is they've taken insulin and they've engineered microorganisms like bacteria to produce insulin itself. So synthetic biology, Patrick and I argue quite a bit about what it really is, and a lot of people do. It's an umbrella term for a lot of different fields. So for example, I have a list right here of a bunch of many different applications that can be covered under. Metabolic engineering, for example, is something that our lab focuses on quite a bit, and that's mainly my expertise. So that would be looking at the metabolism, which is all the reactions and all of the chemicals in a cell, and how can you change that metabolism to make some kind of product, some kind of chemical product. You can also use microorganisms and synthetic biology for bioremediation, for example, cleaning up wastewater pools from mining. Patrick's going to talk a bit about that. And of course, everyone thinks about health when they think about biology. This is very critical. For example, in the human gut microbiome, there's a lot of stuff that we don't understand that relates just the bacteria in your gut to everything in your body. Traditionally also, in terms of industrial fermentation, everyone knows that, for example, alcohol and beer has been made with yeast, and that's been done for thousands and thousands of years. But a very new thing, as some of you may have heard of, is that people are, for example, engineering yeast to make cannabinoids now. So those are made from plants normally, but people are taking the genes from these plants and putting them into microorganisms to make them more efficiently. Also on the docket, you can use synthetic biology to make biomaterials and renewable energy. And more of a niche thing that's very futuresque would be information storage. So this little video, this little GIF of a horse was made by the George Church Lab. So they took this video on the left and they coded it into DNA and they put it into bacteria. And when they replay it back, there's not many errors, and you can actually show that you can store video in DNA. And this information is stable for hundreds of thousands of years, which is crazy when you think about it. So it's not ready for commercial applications yet, but it's very futuristic. So as I was saying, metabolic engineering is kind of like what I focus on in particular. And the definition of metabolic engineering would be to change the cells inside the resources, so the proteins it's expressing, to produce a useful metabolic product. When you think of a metabolic product, this would be some kind of chemical. So you could draw the organic structure with stick figure diagrams like we have up here in the top right. It's not a protein. So we had said we can make proteins before, but proteins are not metabolic products. One thing about metabolic engineering I find is you have to wear a lot of hats with metabolic engineering. And what I've kind of outlined here is the traditional process of metabolically engineering an organism, a microorganism. First, you would start with metabolic modeling, which is you can model all of the reactions inside of a cell and all of the chemicals inside of a cell using linear algebra and programming, such as with Python. to find whatever knockouts or gene deletions you want to make. Then you would go into the lab and you would do genetic engineering. So you would see the genes that you need to delete or edit, and you would go and do genome editing to get rid of those genes or add genes, and maybe add genes from organisms that have the proteins that you want to express. And then there's a little bit of the engineering side that comes in. So you would get those bacteria or yeast into a reactor, and you would make them grow. And basically, as they grow, they would make whatever product you're interested in. And this has a lot of process control. So you're working with big reactors. Our lab usually uses fairly small reactors, but in industry, the reactors are hundreds of thousands of liters, which is very cool, honestly. And then there's also the chemistry side and statistics. So you have to ensure that your organisms are making what you want. So you have to quantify it using some advanced chemistry techniques, such as, you'll learn about these in undergraduate classes, but like HPLC. Yeah. So one example of metabolic engineering would have been what my research was for my master's, adipic acid. So adipic acid is this chemical here, and it's a precursor to make nylon. So nylon is used to make a lot of your clothes and different textile materials. It's very important. Traditionally, what you do is you take a petrochemical feedstock. So it's mainly cyclohexane. And we know that petrochemicals are not sustainable, and they have other issues with greenhouse gas emissions. But not only this, you have to oxidize it using nitric acid. And this releases nitric oxides, which are very potent greenhouse gases. So there's many issues with traditional adipic acid production. However, people have been thinking about making adipic acid, which is not made by microorganisms at all, using microbes. So if you take a bunch of different and you genetically engineer a microorganism such as E. coli, you can actually force your bacteria to make something that doesn't exist in nature. And so that's what my research focused on. You start with a carbohydrate. I use glucose, but in the industry, you would use something like corn syrup. And then you feed it to your engineered GMO bacteria or yeast, and they would make your product, so adipic acid.
Okay, so we're going to switch gears a bit. I want to talk about bioremediation. So to start it off, you're probably familiar with what salmonella is. Salmonella infections lead to food poisoning, typically from mishandled poultry, so chicken. And how salmonella works and how these kind of food poisoning events happen is these bacteria will get into your GI tract, your gastrointestinal tract. Now, the issue is that for these bacteria to survive in your stomach, in your intestines, which is mostly acidic, the bacteria have to do something about the acidity. And so they have special proteins, which we talked about before. These proteins are able to help kind of neutralize inside the cell. So instead of it being acidic around the cell, which would kill the cell, it expresses it and it creates these proteins that help bring the pH back towards 7, which is a good thing. Now, these proteins that do this job, they require nickel. And nickel isn't something that's very common in your diet. So what these bacteria have done over time, over thousands of years, is they've evolved specific protein systems that are incredibly specific for nickel. They will scavenge nickel from the lowest concentrations and bring it into the cells and incorporate into these proteins so that it's able to help fight off the acidity, basically. Now, where else do we find nickel in the environment? If you look into the mining industry, it turns out that one of the biggest types of mining, called hydrometallurgy, it uses acids to basically melt rocks under high heat and pressure. And you want to melt the rock so that you can get to the valuable metals. So for example, gold, silver, and nickel as well. Now, doing this process, you generate literally millions of liters of wastewater in the form of liquid mine tailings, and it's acidic. Metal is dissolved in acid, acidic water. And what happens is that because it's acidic, all the metal from the previous process is stuck inside the water. And when the water is put back into the environment, as you can see in the picture here, there's metal in the environment too, especially iron. So the acidity frees up the iron and puts it back into solution, which is why you see this orange color. Now, the question I have is, how do you re-engineer bacteria or biology in general to deal with the heavy mineral contamination you find in wastewaters like this? So in synthetic biology, there's a process called enhanced bioaccumulation. The idea is that these proteins that you find in food poisoning bacteria, you take just these protein systems that are really specific for nickel, that are very good at scavenging nickel, and you put it into bacteria of choice. And then with these bacteria, you redesign them to mass produce these systems so that you can absorb the metal from the wastewater from outside the cell into the cell. And then you also equip the bacteria with storage proteins or storage systems, for example, that you can see on the bottom over here. And what this will do is allow the cell to now accumulate the metal inside the cell and take it out of the wastewater and store it inside itself. And then you separate the bacteria afterwards and process it later on. Something else I want to talk about is the health applications of synthetic biology. So for synthetic biology, you can produce therapeutics. You can produce proteins that are beneficial for humans that will serve as medicine for disease. What you can also do with synthetic biology is use it as a diagnostic for disease. Rather than treating it, you're trying to prevent it or detect it. So back in 2016, the Zika virus is a very big phenomenon over in Brazil and South America. And what the Zika virus would do is not much to the infected person besides the fever, but when the Zika virus was passed on from a mother to a child through like an infant, it would cause birth defects, as you can see on the right side. So one of the labs that we have at U of T, the professor basically created a paper-based diagnostic, and I'll kind of walk you through it really briefly. If you think back to DNA, DNA has this kind of spirally structure, and you can again see that over here. The difference here that you see on the screen is that it's spirally, but it's artificially designed by humans to react with the DNA, with the RNA of these viruses. So on the far left, you see over here, this is where the computational biologist, the person who has a mathematical, computer science, and a biology understanding, they combine their knowledge and they design pieces of DNA or RNA that they will then put onto this paper disk. What this DNA and RNA will do is that once you store it and then you re-expose it to a sample from a human that might have viral DNA or RNA inside of it, this piece of DNA will react with that RNA and then cause a color change from yellow to purple. And this color change is pretty much 0 or 100. If you see purple, it means that the disease is present. And so this is one way that synthetic biology can be used as kind of a diagnostic tool for health and disease. And so this kind of summarizes our presentation. What we really want to emphasize in this short presentation is that in synthetic biology, because it includes so many different fields, there's a lot of opportunity to enter this field, to use biology to engineer solutions for our future problems and today's problems. So it's highly interdisciplinary. You don't have to go through one specific linear path to get to synthetic biology. A lot of careers do have a very linear path, and that's completely fine. Synthetic biology is one of those careers where your path is not going to be linear. There is no specific bachelor's degree in Canada that will bring you towards synthetic biology. And that's because synthetic biology, you can approach it from a biochemistry perspective, from an engineering, like a chemical engineering perspective. You could take electrical engineering as well and then enter synthetic biology as well. And it doesn't necessarily have to be science and engineering to get into synthetic biology, because synthetic biology nowadays heavily embraces people who have a social sciences and a humanities background to help us understand what are the ethics behind what we're doing. You know, because we're producing GMOs, we're engineering life. There's a lot of ethical implications to engineering life, to modifying how the DNA looks like for these organisms. And so we need people to specifically develop policies that kind of control, I guess, scientists and engineers, honestly, and what they can do, what they cannot do. And lastly, for the people who are especially interested in economics and business, synthetic biology has huge potential to really impact the market. And so understanding the business behind science and engineering, this is an incredibly important thing for synthetic biology. And that pretty much summarizes the presentation. Scott, is there anything you want to add?
No, you said it perfectly.
Sweet. So then we're opening up for questions. We welcome all sorts of questions on synthetic biology. If you have questions about like just general career advice too, we're also open to that. Thank you.
That's great. Thank you, guys. I'm just going to remind classrooms that if you have questions that you want to submit, open the Q&A window and you can type them in there and I'll relay them to our guests. I had a couple of things that I wanted to just kind of double back on. Scott, you talked about your work with new nylon development.